everyone, thank you so much for clicking onto the video. So the first learning objective is to look at the structures of the phosphate ion 2-deoxyribose and the 4 bases adenine, cytosine, guanine and thymine, which are all given in the chemistry data book. We're then going to look at how a nucleotide is made up from a phosphate ion bonded to 2-deoxyribose, which is in turn bonded to one of the 4 bases. And then look at how a single strand of DNA is a polymer of nucleotides linked by covalent bonds between the phosphate group of one nucleotide and the 2-deoxyribose of another nucleotide, and how this results in a sugar-phosphate polymer chain with bases attached to the sugars in the chain. We're then going to look at how DNA exists as 2 complementary strands arranged in the form of a double helix. So by the end, we should be able to explain how hydrogen bonding between base pairs leads to the 2 complementary strands of DNA. So now we're going to look at nucleotides. So DNA, or deoxyribonucleic acid, is a polymer. And so it's made up of 4 different monomers. And it's the way in which all these monomers are arranged that give us the huge variety of different products. So the monomers which make up DNA are called nucleotides. And a nucleotide is made up of 3 parts. We have the phosphate, the sugar, and also the base. And there are 4 different bases that we can use: cytosine, thymine, adenine, and guanine. And all of these can be shortened down to just one letter, so C, T, A, and G. Adenine and guanine are collectively known as purines, and cytosine and thymine are collectively known as pyrimidines. And they're all called bases because they have nitrogen atoms which have a lone pair of electrons which can accept a proton. And that's the definition of a Bronsted-Lowry base. So in your data booklet, you should have these molecules. You should have the phosphate and sugars and also the bases, because you'll be asked to draw out the nucleotides. So on the right, we have a general structure of a nucleotide. So what we have is, at the top, we have a phosphate, and this is bonded to the deoxyribose sugar, and then we have a base which is attached onto this deoxyribose sugar. So for our example, we're going to look at drawing the nucleotide which contains the base cytosine. So first what we need to do is use the phosphate. So the phosphate goes on the top. So we have an OH group, and then we have a phosphorus, and then we have a double bond oxygen. And one thing to know is that they've drawn this as an ion, but we actually need the phosphate without it being an ion, so being neutral. So this oxygen actually has a hydrogen on it. And then I'm going to draw an oxygen. And this is because when we bond phosphate and deoxyribose sugar together, we eliminate a water molecule. And so this is why I'm just drawing the oxygen. If we draw out 2-deoxyribose in its displayed formula, then we should be able to see what's going on a bit more. So we have a carbon with the 2 hydrogens, and then we have an OH group attached onto the carbon. Then we have another carbon and a hydrogen, and then we have the rest of this molecule. So we have an oxygen, and then another carbon, and an OH and a hydrogen, and then we have this bonded to another carbon and another carbon here, and then we have an OH on this carbon, a hydrogen here, and 2 hydrogens on this carbon. So this is our 2-deoxyribose, but in displayed formula. And so what we've done is we've removed a molecule of water. So we've got rid of a hydrogen here and also the OH here. And it doesn't matter how you do it, so you can remove the OH off the phosphate and just the hydrogen off this OH in the 2-deoxyribose. It doesn't matter as long as you're just removing a molecule of water. And so that's why I have an oxygen here that I've written for the phosphate. And now I'm going to join on this 2-deoxyribose. So this oxygen will then be attached to a carbon, and then we would have 2 hydrogens, and then another carbon, and then we'd have the hydrogen, and now we'd have the rest of this 2-deoxyribose. So we have an oxygen here, a carbon, and then 2 more carbons here. So we'd have an OH, a hydrogen, so all of this bit doesn't change. And then for this carbon, I'm just going to leave it as it is for now, and not have the OH, because we're going to be joining the base onto this carbon. So again, we eliminate another molecule of water. So we're going to be using cytosine. So again, to make it a bit easier, I'm going to draw cytosine in its displayed formula. So we have a carbon, another carbon, we have an NH2 group on this carbon, and then we have a double bond to a nitrogen, another carbon, double bond oxygen, then we have a nitrogen, a hydrogen, and then a carbon, and then we have a double bond to this carbon, so we must have a hydrogen here and also a hydrogen here. So this is our cytosine. So what we do is when we bond this cytosine to the 2-deoxyribose, we have to eliminate a molecule of water. And so what we do is we get rid of this OH here and a hydrogen off this nitrogen. And this is how we bond it. So now this carbon off the 2-deoxyribose will be bonded with a nitrogen. And now we need to kind of rotate this cytosine around. And so what we get is we have a carbon here, double bond oxygen, and it still has to be hexagonal shaped. So we have a nitrogen here, and then a double bond to a carbon, NH2 group, and then we have a carbon at the top, and then this is attached to a hydrogen, and then we have carbon here, and this is singly bonded to a nitrogen, and we have a hydrogen here. So this would be our nucleotide. So what we have is that this purple bit here is the base, and then we have the blue bit, which is this sugar, which is 2-deoxyribose, and then the green bit is the phosphate. So when nucleotides add together to form DNA, we form what's called a phosphate-sugar backbone. So we have a phosphate and then a sugar underneath, and then we have another phosphate and then another sugar, and it carries on. And so what we do is this OH here will react with this OH of another sugar. So not this sugar, but another sugar. And this is how we form this phosphate-sugar backbone. And so this is how we can get more and more nucleotides to add on to form a polymer chain. And it's the arrangement of the bases along this chain, which may be in any order, which leads to the variety of DNA. So now we're going to talk about this phosphate-sugar backbone. Many nucleotides are linked together by a reaction between the OH group of a phosphate on one nucleotide and the OH group of a sugar on another nucleotide. So the sugar is the 2-deoxyribose. And this eliminates a water molecule. And so because we're adding two things together and eliminating a small molecule, this is an example of condensation polymerization. And what we formed are covalent bonds between the phosphate of one nucleotide and the sugar of the other nucleotide. And this forms a polymer chain which has a backbone made up of phosphate and sugar with the bases attached. And so this is why we call it the phosphate-sugar backbone. And the arrangement of the bases along this chain leads to the variety of DNA that we get. And the DNA molecules can be specified by the order of their bases. So for example, we could have the C stand for cytosine, so we could have CCAG, TCAG, and so on. Cells in the body can read the sequences of bases to produce proteins. And 3 bases are the code for one amino acid. And we also know this as a triplet code. However, chemists can now read the sequence of bases taken from living things, including humans, and this is a process we call DNA sequencing. So here we have a diagram of this phosphate-sugar backbone. So before we looked at just a section, this nucleotide here, this is what we looked at before. And when we add loads of these sugars with phosphates, this long chain here is what we're talking about when we say the phosphate sugar backbone. Because basically we have lots of phosphates and sugars alternating, and they're all covalently bonded together. So all of these are covalent bonds. And then we have the bases, which are attached to all the sugars. And so we have an order of bases, a specific order. So for this example, we have the order of A, G, T, C, A, G. So this would be our DNA strand, the order of the bases in this DNA strand. So for example, if we were told to draw how two nucleotides with cytosine bases join together, what we would do would be first draw out the two nucleotides with the cytosine bases. So what we have is OH, and then we have a phosphorus, double bond oxygen, and then an OH attached to it on the other side. And then we have an oxygen, a CH2, and then a carbon, a hydrogen, and then on this carbon we have the oxygen and the other carbon, and then we have these two carbons here, this one with an OH, and this one with a hydrogen, another hydrogen, and a hydrogen, and then this carbon has a hydrogen on it, and it's then bonded to this cytosine. Which we can get from the data booklet. Then we have a hydrogen on here, and a hydrogen on here, and an amine group on this carbon. And then now we can draw the same nucleotide. So I'm going to draw this a bit underneath. So what we have is, again, we have an OH, and then a phosphorus, double bond oxygen, and then on the other side we have OH, and then an oxygen, a CH2, then a carbon, and then we have the hydrogen again, and then this now has the oxygen and another carbon, and then two carbons here. So we have a hydrogen and an OH, and then a hydrogen and another one, and then this carbon has a hydrogen, and again this is bonded to our cytosine. So these are our two nucleotides. And for this example, we're using the same one. So we're using two of these cytosine nucleotides. And so we've seen before that this is the OH which reacts, and also this one. So this is the same on this second nucleotide as well. So to join these two nucleotides together, what we do is we form a bond between this sugar and this phosphate. And so we remove this water. And then the stars here and here just indicate that if we were to bond more nucleotides together, these are the species that would be reacting with the other sections of the phosphate and sugar. So this OH at the bottom here would react with another phosphate, so another hydrogen off a phosphate, to remove the water and form another covalent bond between this nucleotide and the other nucleotide. So these two stars just indicate that they can react with more nucleotides to continue this phosphate sugar backbone. And so what we get is we get the joining of these two nucleotides. So we have OH, and I'm going to put a star here because this is what will react with another nucleotide. And then we have phosphorus, double bond oxygen, an OH, sorry, and an oxygen, and a CH2, and a carbon, and a hydrogen, oxygen, and then we've got the carbon, another carbon, and then this is bonded to our cytosine. And then we have a hydrogen, and 2 here, and also 1 here. So now we formed a bond with the other phosphate using this sugar, this 2-deoxyribose. So this carbon now is attached to an oxygen. So we have an oxygen, and then we have the rest of the phosphate. And so again, we have the rest of this nucleotide. So we now have an OH here, and again I'm going to put a star because this is what will react with another phosphate to continue this backbone. And then a carbon, and the 2 hydrogens, a carbon, and then this is again attached to a cytosine. So this is our completed joining of 2 nucleotides. And this is what we've ended up with. So we've started off with the 2 nucleotides which are separate, and we've removed this water, and so we've gone through condensation polymerization, and now we've formed this phosphate sugar backbone. And the reason we call it that is because we have a phosphate, which is this general group, or it's just with the oxygen. So this is your phosphate, and then we have a sugar here, and then another phosphate. There should be an oxygen there actually. We need an oxygen in between this phosphorus and CH2. So we have an oxygen, and then CH2, and then the carbon. And then this bit is our phosphate, and then sugar. So this is why we call it a phosphate sugar backbone. Now we're going to look at the double helix. So DNA exists as 2 strands, the strands that we've seen before, so the phosphate sugar backbones, and these are held together by hydrogen bonding. The bases adenine and thymine can hydrogen bond together, as can guanine and cytosine. And a really easy way to remember this is that pointy letters go together and curly letters go together. So we have adenine and thymine, and these are both kind of pointy letters. And we have cytosine and guanine, which are both kind of curly letters. And so we have the rule that pointy letters can hydrogen bond together, and the curly letters will also hydrogen bond together. But for example, we can't have adenine hydrogen bonding with guanine because they can't hydrogen bond with each other. And this means that the 2 DNA molecules or strands can bond together to form a pair, but only if their bases are in a complementary order. So for an example of that would be if we had a strand that looked like this. So if we had a strand which had adenine and then thymine, and cytosine, and then a guanine, and then another thymine and an adenine. And if in the other strand we had a cytosine, a guanine, and then an adenine and another adenine and a thymine, these two strands would not be complementary to each other, because if we have a look at adenine and cytosine, we can't form a hydrogen bond. If you look at thymine and guanine, we can't form a hydrogen bond there either. Cytosine and guanine can form a hydrogen bond, so we do get some hydrogen bonding there. Adenine and guanine don't have hydrogen bonding again, and then we get some hydrogen bonding here and here also. But we need every single one to be able to form a hydrogen bond, so all of the bases. And so these two strands are not complementary. And the shapes of the molecules are such that in order to fit neatly together, the two strands wind around each other in the shape of a helix. So this here is our helix shape. Now we're going to talk a bit more about hydrogen bonding between the bases. So we know that pointy letters hydrogen bond together and curly letters hydrogen bond together. So from that we know that adenine and thymine hydrogen bond together, and guanine and cytosine bond together. So first let's look at thymine and adenine. So I'm going to draw thymine first, and we have carbon, so I'm just going to copy these skeletal formulas. So we have this NH here, and then we have a carbon and then an NH at the bottom, and then a double bond oxygen here, and then we have a carbon going towards the nitrogen, we have a double bond oxygen, and then a methyl group here. So this is our thymine. And then for adenine, we have a nitrogen and then a double bond to a carbon and then to another carbon, and we have a carbon here, and this is also a double bond, a carbon here, and a double bond, and then we have a nitrogen at the bottom, and we have a carbon here. And then we have the other side of it, so this goes to an NH, and then we have this section where we have a nitrogen, and then this is a double bond to this carbon. And then we have an amine group at the top. So this is our adenine. And how this hydrogen bonds is that, first of all, we need to remember that hydrogen bonds form between really electronegative atoms and hydrogen. And so the really electronegative atoms that we have, which form hydrogen bonds with hydrogen, are either fluorine, oxygen, or nitrogen. And so thymine and adenine, they form hydrogen bonds from hydrogens to the oxygen and to the nitrogen, because we don't have any fluorines. So first what we have is this oxygen bonds to one of the hydrogens from this NH2. So if I draw out this NH2, we have N and then two hydrogens here. And so we form a hydrogen bond with one of these hydrogens. Let's say this one. And so this is our hydrogen bond. And then the other hydrogen bond we form is between this hydrogen and this nitrogen. So this is also a hydrogen bond. And so from this, we can see that adenine and thymine, this is really important to remember, adenine and thymine form two hydrogen bonds. And it's really important to remember that they only form two. And another thing to note is that this nitrogen here and this nitrogen here, these are the ones which form bonds with the sugar. so the 2-deoxyribose. And what I mean by that is that if we remember back to the general structure of a nucleotide, we have a phosphate, which is then attached to a sugar. And then on the sugar, we then have attached this base. So we have the base here, and then we have the sugar, and then we have the phosphate. So what I mean by these nitrogens are that these are the nitrogens which form the covalent bond with the carbon of the 2-deoxyribose, because these are part of 2 nucleotides. So what we have is we have this. This would be the phosphate sugar backbone, and then we would have bases attached to this. So we would have C, for example, and then A, and then G, and then say we have another A. And then on the other side, we would have a G, because we know that curly letters go together. So this has to be a guanine, and then we have a thymine, a cytosine, and another guanine. And so this would be our complementary strand. And so the one we're looking at here is this adenine and thymine. So this has to be a thymine. So right now the example we're looking at is between adenine and thymine. So this is hydrogen bonding that we're looking at. So the blue lines here are the phosphate sugar backbones. So we're taking 2 strands and hydrogen bonding them together. And secondly, let's look at the hydrogen bonding between cytosine and guanine. So this time, I'm just going to draw out the molecules again. What we have is, first, let's draw out cytosine. So we have a bond here and then NH2. And then we have another carbon to a nitrogen. And then we have a bond to a carbon, and then double bond here. And then we have a nitrogen at the bottom, and then a H, a hydrogen, and then a double bond O. So this is our cytosine. And then we're bonding this to guanine. So we have NH here, and then we have up to a carbon, and then here's a double bond. Then we have a carbon here and a double bond. Then we have a nitrogen, and this goes up to here. And then we have the other side. So we have a nitrogen, and then we have a carbon here, and then a hydrogen, a nitrogen, sorry, and then a hydrogen, and this connector. So you can just copy these from the data booklet. And we have double bond oxygen and an NH2 here. So this is our guanine. So the hydrogen bonds which form here are that we have this hydrogen which bonds to this oxygen. And then we also have this nitrogen and this hydrogen. And then finally we have this oxygen and this hydrogen. And so we can see that cytosine and guanine form 3 hydrogen bonds. So you need to remember that adenine and thymine form 2 hydrogen bonds with each other, and cytosine and guanine form 3 hydrogen bonds with each other. And the nitrogens which are attached to the sugars are this one and also this one. And something really useful to help you remember which elements form hydrogen bonds is that looking back at the data booklet, we can highlight all the different elements which form hydrogen bonds. So for adenine, the bonds which form are one of these hydrogens and then also this nitrogen. And when we look at thymine, so when we bond this with thymine, we bond it with this top oxygen and this hydrogen. And so you only need to remember that we need to bond it in the other direction. So we have thymine on the left and then adenine on the right. So that should help you. And then for guanine and cytosine, guanine bonds using this oxygen, this hydrogen, and one of these hydrogens. And then cytosine bonds using one of these hydrogens, this nitrogen, and this oxygen. So if you remember that, then this becomes really easy because if you write cytosine on the left and then have guanine on the right, then you can just form these hydrogen bonds from all these different atoms. So it's really useful to just remember which atoms are used in the hydrogen bonding for each of these bases. So now we're going to talk about cell division. DNA holds the instructions for making proteins, and these proteins are important chemicals in living organisms. DNA is present in every cell, and when the cell divides, the DNA must be copied exactly. So what happens is that firstly, the hydrogen bonds between the bases are broken, and the double helix of the DNA then starts to unravel. And a really important thing to know is that even though the hydrogen bonds break, the two strands of DNA will remain intact. And this is because hydrogen bonds have only 10% of the strength of covalent bonds. And so we can break these weaker hydrogen bonds without breaking the covalent bonds of the DNA chain. And so we retain the sequence of bases that we have originally on the chain. The second step is that a cell contains a mixture of separate nucleotides. And this is present in every cell. And these will then move in and pair up with the newly exposed bases. Then what happens is that the nucleotides then link up via the phosphate sugar bonding. So these are the covalent bonds. And now we've formed two double helix molecules which are identical. And this is why this process is called replication. So we're going to quickly draw some diagrams. So firstly, we have the strand of DNA. So we have one strand. So the double helix is made out of two strands. So this is the first strand, and we're going to have an adenine, a thymine, a cytosine, and a guanine, and it just continues on like that. So this is just an example. And so that means that on this complementary strand, we would have to have a thymine, an adenine, a guanine, and a cytosine. And we also have hydrogen bonding between all of these bases. So this is our weak hydrogen bonding. And in this cell, we also have separate nucleotides. So let's say we have an adenine here, and then we could have a cytosine and a thymine and a guanine, another adenine. So this is just a random mixture that we have of these separate nucleotides. And so what happens first is that the two strands will separate because we're breaking these weak hydrogen bonds. And so we have that the first strand where we had adenine, thymine, guanine, and cytosine, this will break off from this other strand where we had thymine, and then adenine, cytosine, and then guanine. So they've now broken off from each other. and they still have the same mixture of separate nucleotides in this cell. So we still have these little separate nucleotides. And so what happens now is that we can form hydrogen bonds. These nucleotides move in to the bases which have now been exposed, and we can form hydrogen bonds between the bases. So what we get is that we have one strand here where we originally had adenine, and then thymine, and then cytosine, and then guanine, and then cytosine. And so the complementary bases would be a thymine, an adenine, a cytosine, and a guanine. And so these would all be separate nucleotides, and that's something really important to remember. And then for the other strand, we had thymine, and then adenine, cytosine, and then guanine. And we also have to remember that these phosphate sugar backbones aren't broken. Because they're held together by really strong covalent bonding. And then the complementary bases for these would be adenine, thymine, guanine, and cytosine. So again, these would be separate nucleotides. And the final step would be bonding these separate nucleotides by joining them up using covalent bonding. So to make this phosphate sugar backbone. So we react the phosphate with the sugars. So what we get is we have this original strand here of adenine, thymine, guanine, and cytosine. And then we have a complementary strand here. Actually, on the third diagram, we should have hydrogen bonding between these bases, because that's how we've managed to put them into their positions, is by hydrogen bonding with the newly exposed bases. And so in this last step, we then have just all of these separate nucleotides joined on together with these hydrogen bonds between the bases like we had in the previous step. And then for the other one, what we had was thymine, and then adenine, and then cytosine, and then guanine. And then for the complementary, we have adenine, thymine, guanine, and cytosine. And again, we have hydrogen bonding between all the bases. So now what we formed are two identical double helixes of DNA. So these are absolutely identical. So they're identical double helixes. And so what we've done is replicated the DNA, which basically just means that we've copied it exactly. And so what we've done is first we started off with this DNA strand, this DNA double helix, which is made out of two strands. So this section here is one strand, and this is a complementary strand. And we have the separate nucleotides in the mixture with these two strands. And then what happens first is that the weak hydrogen bonding between the bases of this double helix then break. And so we have the helix of the DNA unraveling. And then for step three, the separate nucleotides inside the cell, so the mixture, then move towards their complementary bases, which have now been exposed. So we form hydrogen bonds between these bases. And then the final step is where we have the phosphate sugar backbone forming. So the covalent bonds form between the phosphates and the sugars of different separate nucleotides. And so this is how we've replicated the DNA.